Okay, fertilization. Let's just go over the diagram. Here we have the ovary, mesoverian ligaments, okay, that holds the ovary to the walls of the abdomen, and the infundibulum. This is the oviduct or fallopian tube, if you will. This is the uterus, where there are two sets of muscles. They're called myometrium, and these, the ones that are on the cortex or the peripheral region, known as the endometrium. They are the ones which get ready to receive. It forms a structure like that in order to receive a fertilized egg. Okay? So this preparation of the uterus is done only after or only if the egg cell is fertilized. So let's go over how this fertilization occurs. Now we understand that the ovaries work alternately every 28 days and it releases a mature egg cell. The egg cell is not alone. We understand that the egg cell is a large cell surrounded by supportive cells known as corona radiata. And it also has a structure called the zona pellucida. Okay? And this is the membrane, the actual membrane of the egg cell. And it contains 22 plus X. It can contribute only X chromosome. So here is this cell moving on. And this moves forward because of peristalsis, which is the wave-like action of the oviduct. Okay? It is immotile, so it needs to be propelled or pushed forward by the walls of the oviduct. This finger-like projection that you see is called the fimbriate or infundibulum. Its job is to grab hold of the released egg and drive it in. So it takes around 7 days for the egg cell to move from the infundibulum to the uterus. So during this time, as it passes through the oviduct, its sperm cells are released into the female reproductive structure. Then they will reach the egg cells and fertilize it. Now, every time the sperm cells are released, there are around millions, more than 30, let's say 100 million sperm cells are released. Why so much of sperm cells are released? This is a way in which nature tries to take the best, the most healthiest sperm cell in order to reach the egg cell and fertilize it. So it's only the healthiest cell which eventually goes in, reaches the egg cell. Most of these sperm cells die in the acidic condition of the female reproductive tract. Now, if the cells are if the sperm cells reach the egg cell, it injects its nucleus, and that is called fertilization. So this is what basically fertilization is. But there's a little more to it. We talked about how this zona pellucida is a structure which actually prevents polyspermy. Polyspermy meaning the fertilization of or the entry of more than one set of nuclei into the egg cell. Okay? And it also prevents the sperms of another species from injecting its genetic material into the egg cell. So we talked about how this zona pellucida is species specific. Just want to talk about this zona pellucida a little bit. Zona pellucida is actually a glycoprotein, meaning it is a protein containing some amount of glucose. Okay, glycoprotein. We understand from last year's biomolecules that proteins are long chains, and they do not stay like that. They are given a special structure. So they form a structure like that, and they always have binding sites So in this case, the binding sites are made of sugar. This glucose part forms the binding site. So these are the places where a sperm cell can latch on to the zona pellucida. Okay? So when the sperm cells of the same species, or human sperm cells, are released, they come and they can latch on to these binding sites of the zona pellucida. And once it does that, it ignites or it stimulates the acrosome to release its enzyme. Now we understand that the enzyme that's present there in the acrosome is called hyaluronidase. So this enzyme is going to dissolve the zona pellucida and everything that's here, and lets the sperm cell really enter this wall, through the wall. So it reaches the cell membrane. So this pink line that I've drawn here, double line, that is the egg plasma membrane. Okay? Just under it, or just inside it, I'm going to draw these here. These round structures that are on the periphery of towards the skin towards the cell membrane of the egg cell. These are called your cortical granules. They contain some enzymes, and these granules, which has a very important role to play here. So once it injects its nucleus into the egg cell, so this is the 22 plus, let's say, X chromosome. Once it injects its nucleus into it, this stimulates, or it triggers off what's called the cortical reaction. Okay? Cortical reaction, which is basically the rupturing up of these granules that you see here, the organelles called cortical granules. They rupture up and they undergo what is called exocytosis. I'm just going to bring exocytosis here a little bit. Okay? Exocytosis actually means when a cell organelle reaches the cell membrane, it fuses with it, and there's a break. Okay, and then it can release its content. So here it's releasing its content, just like amoeba, which is trying to throw out its waste. The waste goes and reaches the membrane, it touches the membrane, and the membrane diffuses or it dissolves, and then it helps release. So this is known as exocytosis. So exocytosis occurs here, and cortical cells releases its content. Okay, and we have these substances that are released by the cortical cells, and there is a tiny space in between the plasma membrane and the zona pellucida. Just under the zona pellucida, you find what's called the vitaline layer, and in between you find a tiny space known as the I've not mentioned it here, but it's called the perivitaline space. It's called perivitaline space. So this perivitaline space is filled up with the granules secreted by the cortical granules. So this space is going to be filled up like that. Since it's granules, it's hard in structure, and it prevents the entry of, the further entry of sperm cells. And it also contains enzymes. This cortical granules also contain enzymes which is released. These enzymes changes the structure of the zona pellucida. Zona pellucida here again. We said the zona pellucida is a protein with binding sites for the sperm cells. Now if an enzyme comes along and changes the structure of the zona pellucida, all these binding sites are broken up. They turn inwards, or just understand that the binding sites are not visible, or it's not there at all. Okay? So in that case, these binding sites are not there at all, meaning even if the sperm cell comes here, it will not be able to latch on to the zona pellucida, and it will not be able to release its acrosome in order to dissolve this particle. So this will persist. Okay? So this is another method in which the further entry of eggs, the head of a sperm cell is prevented. This is called the prevention of polyspermy. Right?